All right, so we're still up here at the top of the mountain, having covered the genetic code, DNA replication, transcription, and translation. And last Monday, coming back from spring break, we were able to talk about viruses. Last Wednesday, we covered the immune system, how the human body and the mammal body in general responds to infection and other threats, including viruses, but also including bacteria and parasites, and in some unfortunate people, peanuts or chocolate or strawberries, because I think the very last thing we might have mentioned would be allergies, which of course is basically an immune reaction to something that's not typically harmful. So I want to talk about something else, talk specifically about mutations today. DNA, of course, is replicated, and back before break, we talked about how that's done. That double helix unzips, and each strand is used as the scaffolding to build a new strand. And so you have two double helices where you formerly had one. The heavy lifting for that is done by a set of enzymes called DNA polymerases. I think earlier I told you it was basically done by little fairies. It's not really done by little fairies, but it is done by specialized enzymes that can basically weld nucleotides into place. There's other enzymes that act on DNA and repair damage to it. DNA is actually a relatively fragile molecule, and there's lots of things that can break it, can break one of the strands, or chemically alter one of the bases or something like that. So the polymerases replicate DNA. There's other sets of enzymes that repair DNA, and they're very, very good. However, they're not perfect. There are some times when they make mistakes. And whenever you make a mistake, you end up with a sequence of DNA with an altered sequence from what it used to be. If you've ever played that game Telephone, it's a little bit like that, where you line up people in a line or maybe in a circle, and one person repeats a sentence to the next person, usually by whispering it. And then the next person whispers it to a third, and the third repeats it to a fourth, and so on. And by the time it goes all the way around, it's usually something completely different from what you started with. It can happen that way with DNA mutation. Whenever you copy a piece of DNA, there is a non-zero chance that one or more of the nucleotides may be altered from what you started with. These can cause a gene to be transcribed and translated into a different protein. These can cause genes to be silenced, no longer transcribed and translated at all. A mutation might put a stop codon in the middle of a gene, and now the gene breaks off in the middle and it no longer is used to transcribe, or can no longer makes a functional protein at all. You can also have mistakes in crossing over that can lead to portions of genes, or sometimes whole genes, getting duplicated or getting deleted. Just as sometimes if you're typing something, sometimes your eye might slip and you might accidentally words out. Accidentally leave words out, or sometimes accidentally duplicate sections. All of these are considered mutations, any kind of change in the sequence of DNA nucleotides that can be passed on to the next generation. Those are all mutations. anything that damages DNA can cause mutations, and a substance that causes mutations is said to be a mutagen. There are certain chemicals that chemically react with DNA and cause it to develop mutations. Ultraviolet light causes particular types of DNA damage that the cell cannot always perfectly repair. Many types of radiation, gamma rays and X-rays can cause damage to DNA. Sometimes that damage can be repaired, other times it's not repaired perfectly. What you're looking at here is a piece of a flat dish. It's called a Petri dish with some agar, with some gel poured in the middle of it. And I think you guys are going to be using this in one of the upcoming labs. And those white specks that are growing on the surface, whoops, sorry. All of these things, all of these white circles are colonies of bacteria. So these are bacteria growing on a culture dish. There is a relatively simple technique that you can grow certain bacteria in a culture dish, expose them to a chemical or anything else, grow the bacteria on the dish, and check for mutations. This particular test happens to be called the Ames test, and it's the first line test for mutagenic activity in new chemicals. If somebody discovers a new, I don't know, fingernail polish or something like that, they'll mix a little bit of it with some bacteria, spread the bacteria on the dish, and they're able to count the number of times that mutations happen. That doesn't always mean that such a compound is perfectly safe for all living things, but bacteria use DNA and they do transcription and translation. At the real nuts and bolts level, they do the same thing that human cells do. And they're affected by the same chemicals and other mutagens that human DNA might be. So a test on bacteria, the Ames test, is the first line test for mutagenic activity. It's the first thing you would do if you had some new, I don't know, food additive or chemical for cosmetics or something like that. You might still have to go on to testing it on animals, I'm afraid, but at least the number of animals can be cut down quite a bit by the use of the Ames test. Right. A reason why this matters is that cells control their division with sets of specialized proteins, and there just is not time to go into the details of how this is done, but you can think of these specialized proteins as being the brakes and the accelerators of cell division. They keep cells from dividing, except at times when they need to divide. There's also this: when a cell is damaged or when a cell is no longer needed in an animal or human body, the cell undergoes a very orderly process of mopping the floor, turning out the lights, putting the chairs up on the tables, and closing down. It's called apoptosis, otherwise known as programmed cell death. And the cell eventually breaks apart into a bunch of tiny little blips that drift away. In your body, the immune system would eventually just mop them up. And this process of orderly shutdown is also controlled by a set of genes that make a certain set of proteins. If you really want to know how it works, Dr. Runge in our department, Dr. Steve Runge, is actually a specialist in apoptosis, programmed cell death. And again, I am skimming over a huge amount of detail, but what you're looking at in this picture is on the right, that's a DNA molecule. Shown here is a computer graphic in blue and red. Binding to it is a green model. That's a computer model of a protein, and you might notice that it's got a lot of alpha helices in its makeup. I just outlined one. What you're looking at is a computer model of a regulatory protein. I don't happen to remember which one it is, but it's a protein that binds to DNA and blocks transcription. So that's the cool thing. You can have genes that are transcribed and translated to make proteins, and the proteins go back to the DNA and bind to them, and some of them will block the transcription of other genes, and we call those repressor proteins, and others of them will promote the transcription of other genes, and we call those promoter proteins. And the upshot is that some genes act as on-off switches for other genes. Some genes turn other genes on and encourage them to be transcribed and translated. Other genes code for proteins that block other genes from getting transcribed or translated. And this is part of how genes can control cellular processes. Some genes code for proteins that promote or repress other genes. And what you're looking at here, please do not try to memorize any details on it whatsoever. But what you're looking at is a small part of the genetic circuitry. This happens to be in animals called sea urchins, which have been used a lot as a model system to study how embryos develop. As you go from a fertilized egg to like 2 cells to 4 cells to 8 to 16 to a ball of cells that develops a gut, on and on and on. As you go through all of these stages, one set of genes may be expressed, they'll be transcribed and translated, and their proteins will switch on or switch off another set of genes, and those genes will switch on or switch off a third set, and those will switch on or switch off a fourth set. And this is what you're looking at in the diagram. Every one of those colored lines shows how one gene either promotes or represses a different gene. And so all of these lines are the ways that the expression of one gene affects the expression of other genes. And you end up with this network of genetic circuitry. Okay, that's a lot of detail. Just know that genes can be transcribed and translated to make proteins, and those proteins can switch on or switch off other genes. Ultimately, that's how you can go from a single fertilized egg to a complex adult body. You're looking there at a mouse embryo. There was a time when you did not look particularly different. This is about what you might have looked like maybe a couple of weeks after conception. And different populations of cells have been stained different colors. And you can see in green, that's the nervous system, and there's quite a system of nerves that's growing through the body. Various other cell populations are stained in purple or red or white. Studying how this works is called developmental biology, and we just have not got time to go into it. It's just an introduction to it would be a pretty advanced upper division course. Let's just say that because genes can influence each other and one gene can promote or shut down other genes, this makes it possible for cells to differentiate. You're made of roughly 200 different cell types. They all came from one fertilized egg. And what's happened is that as that egg divided into more and more and more cells, different groups of cells shut down the genes that weren't relevant to them. Your liver cells still contain the genes that could be used in a nerve cell. Your liver cells still have all of the genes that a nerve cell would need, but those genes have been shut down. They've been deactivated. That's what differentiation means. Cells become more and more specialized as an individual develops from a fertilized egg through an embryo to a mature adult. Okay, and again, that's a huge field, and I can't give you more than the tiniest little taste of it, but that's ultimately how organisms develop from egg to adult. Here's the thing. I started this whole mess by talking about mutations. There are some genes where if they're mutated, they no longer are able to promote or repress the other genes that they're supposed to, and you can get these cascading effects that can end in very severe birth defects. Another reason why this matters is that the control genes that basically determine when a cell divides are called oncogenes. And if those oncogenes are mutated and they no longer function, one possibility is that the cell can begin dividing uncontrollably. You can think of oncogenes as the brakes and accelerators of cell division. In cells that have those oncogenes mutated, they can begin dividing very rapidly, dividing as fast as a car with a stuck accelerator and no brakes. Now, normally when this happens, if a cell starts dividing uncontrollably, the cell hits the self-destruct switch and undergoes apoptosis. Remember that pattern of programmed cell death. The problem is if you have certain mutations will shut off apoptosis, and so you might have a bunch of cells that are dividing rapidly and can't self-destruct, and what you've got there is cancer. Cancer is cell division with the accelerator stuck, the brakes off, and no way to stop the car. It's runaway cell division. And anything that damages DNA is a potential cause of cancer. You may know this word. Substances that increase the risk of cancer are known as carcinogens. That carcino, that's actually Latin for a crab, because ancient Greeks and Romans, they did observe cancer and they thought of it as being like a little crab that gets its claws stuck in your body and won't let go. Yeah, that's why cancer means both a severe disease and the sign of the crab in the zodiac, if anybody was curious. So yeah, anything that damages DNA has the potential for causing cancer if it happens to damage the oncogenes and damage the genes that are involved in apoptosis. And this, by the way, is why you can inherit a susceptibility to certain types of cancer. Women with certain alleles for a gene called BRCA1 have an increased risk of breast cancer. And this is the kind of thing that you can get genetically screened for if you've got a family history of it. And the simple reason is you can inherit oncogenes that are not functioning quite as they should. And that's why some types of cancer run in families. Having said that, not all mutations lead to cancer. There are some mutations that do absolutely nothing. And you might remember that the genetic code that you saw just before break, most amino acids have multiple codons that code for them. So some mutations might change a codon for one amino acid to another codon for the same amino acid, and that does absolutely nothing. Probably the majority of mutations have no effects at all. Even if mutations do change the primary structure and maybe the shape of a protein, sometimes that doesn't affect what the protein does in any significant way. And again, probably the vast majority of mutations are completely neutral. They don't do anything. There are some that can be quite harmful. The mutations that lead to cancer is one example. There are genetic diseases. There's one called Huntington's disease that you can inherit, but there are a reasonable number of people that get Huntington's disease without any family history of it, and it's because the gene involved happens to be susceptible to mutation. But are mutations ever beneficial? Beneficial is a relative thing. You have to look at things in context. But there are mutations, and we've documented them, that are in fact quite beneficial. So some mutations may be very harmful. The majority of mutations don't do anything. And there's others that are beneficial, and this is some. This handsome guy with the beard and the blue eyes is Dr. Richard Lenski at Michigan State. And he has been growing a species of bacterium called Escherichia coli. Now, Escherichia coli is very well understood, and it's very easy to raise. It's been used as a model system for a very long time. And he's been growing Escherichia coli since 1988. And under good conditions, Escherichia coli will divide about 6 times a day. So between 1988 and now, Lenski and his team has watched E. coli for over 76,000 generations. And what he can do is periodically screen his E. coli and look for mutations. This is a lot easier to do with E. coli than it would be with, even with lab rats or something like that. This is a very good model system to look at very general questions. And his finding was that roughly 1 in 12 mutations benefited the bacteria because it gave them increased efficiency in using food. And he was able to trace when and how those mutations happened. He was able to find out exactly what those mutations were. In particular, he found one set of mutations that gave some of his bacteria the ability to use a food source that they previously were not able to make use of. He was growing his bacteria on a sugar called glucose, and some of his bacteria developed the ability to use a food called citrate, which previously they couldn't do. And it's because, be it said, that as his bacteria were growing, in every generation, there were some that had mutations. Some of his bacteria had mutations in the enzymes used to break down food. Most of those mutations probably never did anything. Some of those mutations were probably harmful. And every so often, one of those mutations gave the bacterium the ability to use citrate as a food source. And that's beneficial if there's citrate in the environment that the bacteria live in. And it's not because the bacteria particularly wanted it. Bacteria aren't trying to do anything because bacteria don't have brains. This is a completely mindless process. The gene varies because of mutation. Most of those variants aren't really different. Many of those variants might be harmful, but every now and then, one of them is going to be beneficial. And his estimate was maybe about 1 in 12. Here's another example, 1975. This is a strain of bacteria that for some reason does not have an established specific name. It's called Pinarthrobacter K172. And it was discovered living in wastewater from a factory that produced nylon. And these bacteria were actually capable of digesting nylon byproducts. The joke is that nylon did not exist before 1935. Nylon is a completely artificial fiber using chemicals that, as far as we know, were never produced by any natural process before scientists figured out how to make them. So at some time between 1935 and 1975, bacteria like this living in these wastewater ponds evolved the ability to break down nylon byproducts, and they must have done it by mutation. And it's even been possible to compare the DNA of these bacteria with others and figure out what the mutations are and figure out how they work. That these bacteria, as they were growing in every generation, some of them had mutations. Most of the mutations probably did nothing. Some of the mutations must have been harmful and those bacteria must have died out. But some must have given the bacteria the ability to break down this food supply in their environment that they could not use before. Does this ever happen to people? Well, we don't exactly get this. There is, as far as I know, no mutation that has ever caused a human to shoot optic blasts out of its eyes. But this guy's a mutant. He is a champion cross-country skier. I think he's still alive. He was as of a few years ago, named Eero Montiranta, an athlete from Finland. So here he is at the 1964 Winter Olympics in Innsbruck, Austria. And he's looking pretty happy because he won 2 gold medals and a silver in cross-country skiing. Cross-country skiing is, one of the most vigorous types of exercise that there is, because you've got to pump your arms as well as your legs, and you have to do it over very long distances. It requires enormous amounts of energy to do. And it turned out that Montiranta had a mutation in a gene called EPOR, that's short for erythropoietin receptor. We don't need to worry about what it does, but that mutation caused him to make extra red blood cells. The clinical name for having extra red blood cells is called polycythemia. Don't memorize that term. And yeah, he just made extra red blood cells. This is a pedigree chart, and I can't remember if you're going to do this particular lab or not. But this is a family tree. Squares represent men. Circles represent women. And here's Montiranta right here. Here's our guy. And there's his wife. And they had 3 kids. They had a daughter. And then drawn like that, this is a pair of twins, a boy and a girl. And Montiranta had 3 sisters. And there's Montiranta's dad. The slash right there is indicates that his dad was deceased at the time the study was done. And every one of these simple symbols that is shaded black has extra red blood cells, has the ability to make these extra red blood cells, right? Montiranta's dad passed it on to his oldest daughter and his youngest daughter, as well as Montiranta. His oldest daughter has passed it on to 3 of her daughters, and one of her daughters has a son that's got it, and so on. And we can analyze pedigrees like this to show that this is inherited as a simple Mendelian dominant allele. It follows exactly the same rules as a purple flower color in peas. Some people who have polycythemia are at elevated risk of diseases like stroke. But as far as anybody can tell, Mantiranta's family has never suffered any ill effects. This is him today over here down at the lower right. And there's his wife. And those would be his kids and his nephews in the background. And as far as anybody's been able to tell, his family members don't die young. They don't suffer any especially bad effects, except their blood can transport 50% more oxygen than a typical person's blood. Those of you who follow sports might know that there's a banned process called blood doping, where you take some blood from an athlete, separate the red blood cells and freeze them. And then just before the big game, you inject that athlete with his own red blood cells. And since red blood cells transport oxygen, that means that the athlete's muscles can pick up more oxygen and will function better. That's illegal, but Eero Mantiranta, basically his body does that all the time. He is naturally, perpetually blood doped. And I'm not saying training didn't have a lot to do with it as well, but he does have just a bit of a genetic advantage over everybody else at the Winter Olympics in 1964 because of a mutant allele that he picked up from one of his ancestors and expressed and passed on to some of his kids. And I happen to think this is pretty darn cool, that that's a case of a mutation that we can identify that is actually beneficial, at least for people who want to be champion cross-country skiers. And in case it wasn't clear, the reason why new strains of viruses keep appearing is mutations. The reason why we went from the alpha strain of coronavirus to the beta, to the delta, to the Omicron. Currently, there's concern about a variation of Omicron that they're calling Omicron's little brother. There will probably be more. And like any DNA, virus DNA, or in some cases, virus RNA, is subject to mistakes in copying. Most of these mistakes either do nothing or they've got a negative effect. Most viruses that have a mistake in their DNA or RNA, most of those viruses either aren't changed or they're changed for the worst. But a few enhance the virus's ability to spread and or its ability to dodge the immune system. And the virus doesn't want this to happen because viruses aren't even really alive. Viruses are completely mindless and can't want anything. It's just a fact that viruses vary. And every now and then that random variation caused by mutation just happens to create a variety of virus that is just a bit better at duplicating itself, spreading itself, and or dodging the body's defenses. This is why you have to get a flu shot every year, because there are constantly mutations that create new strains of the flu. Every year, the CDC has to try to predict what the worst strains of flu are going to be in order to develop vaccines in time for the flu season. Sometimes they don't guess right. Some years that flu shot is more effective than others. And the reason why you keep having to have flu shots is that influenza viruses mutate quite a lot. It's also part of the reason why it's been very difficult to develop a vaccine against HIV. HIV mutates so fast that coming up with a vaccine for it is like trying to hit a very rapid moving target. By the time you've developed a vaccine against one strain of HIV, that strain has been replaced by others. Even within one patient's body, that patient may develop multiple new strains of HIV faster than the immune system can adapt. That diagram right there in the center, that's a diagram of COVID. This was the Alpha variant back in 2020. And this thing right there is a diagram of the virus itself. And in red, you can see it's got these spike proteins coming out. Remember, corona means shaped like a crown, and the spike proteins reminded somebody of the pointies on a crown. Surrounding that, that's a representation of the virus's DNA. And there are 3 points there, there, and there, where compared to earlier variants of the COVID-19 virus, the Alpha variant has a deletion, has a nucleotide that's gone missing. And the colored lollipops right there, come on, there, 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 and then all of these over here are points on that virus's DNA where a nucleotide has changed from what its ancestor was. Every time COVID replicated inside somebody's body, there's a chance it would mutate. Most of the time, the mutations either did nothing or they made the virus less effective. But these mutations just so happen to make the virus just a bit more infectious and possibly more resistant to vaccines. One of those mutations changed an amino acid right there, number 501 in the spike protein, from the amino acid asparagine to the amino acid tyrosine. That one little change alters the shape of the spike protein, those spikes coming out of the virus. And that one little change just happens to improve the virus's ability to bind to cell surface proteins. So it makes the virus more infectious, better able to latch onto a host cell and inject its DNA inside. What you're looking at here is a chart of DNA sequences represented by these lines. And everywhere you see a little mark on one of these lines, that represents a mutation relative to the earliest COVID's variety that was introduced. So the beta variety has variations there, and gamma right there. Delta was the really bad one this past, what was it, last Christmas? I forget, time is meaningless now. You can see there's a lot of mutations there. And Omicron has even more. You can see lots of little squares on this line. Those represent points on the virus's DNA where mutations have been detected, and there's quite a lot of them. And Omicron, a lot of those variations affect the spike protein. So right here on the right, this is a diagram of the spike protein of the Omicron variant. And every one of these little flecks of color on the diagram is a point where an amino acid in Omicron came to be different from the corresponding amino acid in the earliest strains of COVID. Every one of those colorful flecks is an amino acid that has changed between December of 2019, when COVID was first isolated, and now. So the spike protein has changed a lot of amino acids, and those changes seem to make it more infectious, although also possibly less severe. You can see there's also mutations, although not quite as many, on the delta spike protein, which is over here. It's that. And I'll say it again, the virus's aren't trying to do this. The viruses have no brains. The viruses can't try to do anything. The viruses are just replicating their DNA because it's what viruses do. And every time they replicate that DNA, there is a chance greater than 0. There is some chance that any given piece of DNA is going to end up with an altered sequence of bases. In other words, it's going to mutate. And the process does not happen with any direction at all. Mutations just happen. I mean, scientists can cause mutations to happen. And in fact, we can cause them very precisely using a technique called CRISPR, which maybe I'll talk about later. But they would happen even if there were no humans at all. These viruses would still mutate. Most of the mutations either don't improve the virus's ability to spread, or they're bad for it. But there is always a chance that one or more of those mutations is actually going to increase the virus's abilities to spread. And those mutations tend to become more and more common because a virus that has them can spread faster than a virus that doesn't have them. And so the viruses aren't trying to do this. Nobody's intending it. It just happens because of mutation, and incidentally, because of natural selection, which we talked about way back in about week 5. And when we reconvene on Wednesday, I will talk more about what natural selection looks like when you couple it with our new understanding of how DNA and genetics work. But for right now, I'm going to stop this recording.